Hello everyone. Today I'm going to share our current study on the feasibility of blood-nerve barrier disruption using focused ultrasound and microbubbles in a rodent model. Focused ultrasound combined with circulating microbubbles has been proved to effectively open the blood-brain barrier and the blood-spinal cord barrier for localized delivery of therapeutic agents. It is non-invasive, targeted, and leading to a reversible permeability change. Its effect on the blood-nerve barrier, however, has not been investigated. Blood-nerve barrier, BNB, or blood-nerve interface, is a physiological boundary between the peripheral nerve axons and the bloodstream that prevents the transfer of substances from the plasma to the nerve fibers. In chronic nerve injuries, the blood-nerve barrier is usually intact, limiting the delivery of therapeutic agents into the injury site. The goal of this study is to investigate the effect of focused ultrasound plus microbubbles on the BNB for potential cell-directed treatment and drug delivery. The animal model used in this study is Thy1 GFP gene-modified rats. The experiment was carried out with a benchtop focused ultrasound system. During preparation, the sciatic nerve on both legs were surgically exposed for focused ultrasound sonication and control. After moving the transducer to the target, Evans blue dye was injected for BNB disruption indication. 5 minutes later, microbubble solution was injected, and ultrasound sonication was delivered simultaneously. 15 to 20 minutes after sonication, compound muscle action potential was measured on both nerves, and the animal was sacrificed afterwards for histology evaluation. Virus peak negative pressure and microbubble dosages were tested. The acoustic emission during sonication were collected with an acoustic detector. Panel A here shows the baseline frequency spectrum before bubble injection, and Panel B shows the frequency spectrum after bubble injection. Ultraharmonic emissions was detected, indicating potential stable cavitation and opening effect. For electrophysiological evaluation, here showing CMAP recordings on the sonicated nerve compared with the contralateral healthy nerve. The pressure used here is 1.2 megapascal. A delayed onset latency was observed on the sonicated nerve, and the CMAP amplitudes were lower, indicating potential change in the nerve conduction activity. However, both body temperature and anesthetic level might affect the readings, so more animals will be required to establish a solid conclusion. When reducing the pressure to 0.3 megapascal, the changes in nerve conduction activity became more subtle. In our ongoing study, we used 0.3 megapascal to avoid tissue damage. When comparing the recordings acquired pre- and 20 minutes post-sonication, no delayed onset latency was observed, but we see a lower CMAP amplitude. The number of animals is still limited, so future work will be carried out to characterize the reversible effect of BNB disruption. The histology evaluation was performed to observe the Evans blue dye leakage corresponding to BNB disruption. The result shown is acquired from an animal sonicated under 0.3 megapascal and a microbubble dosage of 40 microliter per kilogram. The red color showed the Evans blue dye leakage at the target region. In the zoomed-in image, axons are shown as green, and the distribution of Evans blue dye indicated the BNB disruption effect. No evidence of tissue damage was observed on the H&E staining. In conclusion, blood-nerve barrier disruption can be achieved via focused ultrasound combined with microbubbles. Based on our results, 0.3 megapascal plus 40 microliter per kilogram microbubble dose is adequate to introduce BNB disruption without causing tissue damage. Changes in nerve conduction activity were observed on the sonicated nerve, but more work will be done for future investigation. The future work includes investigating MR-guided focused ultrasound sonication to avoid surgery, increasing the number of animals in each group to optimize the focused ultrasound parameters for reversible disruption, and conducting the efficacy study for localized drug delivery into the PNS. Thank you for your attention.